Hello, and yes, this is the one short revision lecture for biopracticals class 12. So let's just, without wasting any time, let's start it quickly. So totally, guys, there will be 7 questions in the whole practical thing. First 2 parts will be of doing an experiment and preparing a slide. We don't have to write anything much. So let's start with the question number 1. In the question number 1, we'll be having 4 experimental choices. So what are those? One is the pollen germination experiment, pollen tube growth on the stigma experiment, transfer section of the ovary, and the nucleic acid staining experiment. So let me just explain you one by one how do we perform the experiments, and then we'll be just moving on further. So yes, the pollen germination experiment. So yeah, we need to show the artificial germination of a pollen grains. We know that the pollen grains usually grow, if this is a stigma, pollen grains are usually going to grow in this direction. But now, in this activity, we are going to do the artificial germination of pollen grains. So let me just start with it. What I need to do is that I need to take Vinca flower. Okay, Vinca flower, let me just, imaginary Vinca flower, it is usually pink in color. Okay, so this is how Vinca flower look, and yeah, we are going to have some stem over here. We are going to have some, something over here. So we need to get the pollen grains right now. In this part of a flower, we will be getting the stigmata, that is the female reproductive part, and in this part of a flower, we will get the male reproductive part. So what you need to do is that you just need to remove all of these petals from your hand, okay, and you will like separately get this thing there. You are separately going to get this, okay, just open this up, and you will see some dust kind of particles, okay, some yellow colored particle, but we do not need that whole yellow color particle because yellow color particle is a anther over there, okay, yellow color particle is an anther over there, so obviously we do not need the whole anther. Some more looks like this, okay, yeah, some dots like this, that is how it looks. Yeah, something like this. So we need the part which is inside that. So what you need to do is that you just touch a brush or the dissecting needle to it and put it inside the, what do we call it as, man? Watch glass. Yeah, we need to put it inside the watch glass. So if this is a watch glass, you're just gonna, no, I'm sorry, man. Wait, I'm sorry about this. Yeah, so once you get the answer, don't do anything right now. What you need to do is that you take a glass slide over here, take a glass slide and put some drops of sucrose solution. Okay, we will be putting some drops of this sucrose solution. Sucrose, it's 10% of sucrose. They might ask you, they might ask this in the viva also. 10% of sucrose will be added over here. In the glass tube, put some drop of sucrose. Okay, consider this to be the sucrose. So now what you need to do is that either use a dissecting needle or the brush. Just take some parts from here. Just touch this anther. Okay, if this is an anther, just touch that anther. Touch this anther. You are gonna get some dust-like particle and put it inside the sucrose solution. Just touch the brush to this anther and dip it inside the sucrose solution. You're automatically gonna get some dust. Okay, and then keep it like that for like 20 to 30 minutes so the germination can happen around. Say, don't be urgency. Don't do this any urgency over there. And then cover it with a cover slip. Cover slip is a very small thing. Okay, it's a very small box type of thing. I hope you all know what is this cover slip. Just cover it with a cover slip and you're ready to go. Place it under the microscope, and show your slide to the examiner. So that contains 3 marks. Okay, your orals are going to get 3 marks if you get that. So now the next will be calculation of the pollen germination. So we need to calculate the numbers of the pollen grains that are germinated. So how would you do it? For example, consider that this is a weave under a microscope. Okay, yes, this is a weave that I'm seeing under a microscope. So what am I able to see? So here I'm able to see some pollen grains like this. Yes, and some of the pollen grains which are already like germinated, and more pollen grains in this way. So yeah, let me draw some over here. So yeah, I can clearly see that there are like 2 pollen grains which are germinated, and 1, 2, 3, 4, 5, 6, 7, 8. Total, there are 8 pollen grains. Okay, so the formula will be N, which is the numbers of the pollen grains that are germinated by the total number of pollen grains into 100, will finally give me the percentage of the pollen germination. So here, if I calculate it in this slide, if the slide somewhat looks like this, it would be like 2 by 1, 2, 3, 4, 5, 6, 7, 8. 2 by 8 into 100. So what's the answer that I'm going to get? Use your calculator. You're obviously taking it right to, 25%. 25% of pollen germination is taking place. This is going to give you 2 marks, and finally 5 marks. You're going to get your 5 marks. This is our first experiment. First experiment in the sense, you're going to get either of the 4 experiments which I'm going to explain you below. We'll see what are all of those. So this is the first experiment that you might get. Okay, moving on to the second experiment. So the name of the second experiment is the pollen tube growth on the stigma experiment. Okay, so what's happening over here? Again, we will be taking our Vinca leaf, not Vinca leaf, what am I saying? Vinca flower. Yeah, we already know it, right? We already know. In Vinca flower, we are going to get this under part will be so small. Okay, it won't be so thick actually. It would be very thin. It will be so thin. Yeah. So this part I said that you're going to get this male reproductive organs, your male reproductive parts, and here you're going to get this female reproductive part. So now use your dissecting needle and just cut open this thing here, and just cut open this thing over here, and you're going to get 5. Yeah, and you're going to get 5 this thing, 5 style stigma and everything. Okay, you're going to take one out of that. Yes, take one out of that. Cut this into half. Cut this into half and place it under a watch glass. Okay, this is a watch glass. Place that thing under a watch glass. Again, add the solution of the cotton blue. Add the cotton blue solution on this. Keep it over there for like 10 to 3 to 5 minutes is enough. We do not need any huge amount of time. Keep it for like 10 to 5 minutes. Then obviously cover it with a glass slip. Yeah, cover it with a cover slip. And you're again good to go, and show it. Okay, show it to your teachers. Report this experiment to your teachers. Yes, you're going to get 3 marks over there once you see there is some germination happening over there. Fine, that's only the thing to be done. So here, 3 marks, you'll get it over that, and the experiment carries are 3 marks. Okay, your performing experiment carries are 3 marks. So yes, the remaining 2 marks, you're going to get it through the neat label diagram. So let me draw this neat label diagram. This is a stigma. And this is a stem. Okay, so there will be some pollen grains over here. This is how it should look under the microscope. Okay, pollen grains, which has done the germination. Naming the whole thing, this part is the stigma, and this is our style. This is the pollen grains. So that's all, and this would give you remaining 2 marks. Okay, yeah, totally you're gonna get this 5 marks if you do only so much. That's all to be done in the second experiment. You might or may not get. Okay, this was the second experiment. Moving on to the next one. Okay, so now next one is the transfer section of an ovary. This again is a very simple one. Let me take this hibiscus. Okay, we all know how is this hibiscus look, right? Somewhat. I mean, ignore my drawing, guys. I'm sorry. Yeah, a hibiscus flower where there is this. We all know it, right? Yeah, we all have all of those, like here, and there. And yeah, we have an ovary over here. These are the petals. These are the sepals. Yeah, these are the calyx, and those are the corolla. So much we know. So what you really need to know in this experiment is that first of all, neatly cut down the petals, cut down all of these petals, okay? Let me use a very bright color. So remove these petals, remove these petals, remove these petals from both the sides. Then you're only gonna get this thing, right? So okay, we are only gonna get that. I'm gonna get this thing. Yeah, this thing is the one which is gonna remain, okay, with all of those, yeah. And this part, we already know that these are the pollen grain part, and this will be the female reproductive part. So this is a dissection of an ovary taking place. So the main important thing that we need is at this part. So how do we get that part? Once you're done with this, the outer layer is completely the male reproductive organ itself. Yeah. So now what you need to do is that you just need to cut open this male reproductive organ. You're going to get a thin white color structure which looks somewhat like this. Okay, thin white color structure which looks somewhat like this. This would be a style, and something at the top, some round type of structure at the top. Okay, which would be the stigma, and then you would get the ovary over here. Okay, this is a male reproductive. You're gonna get the whole male reproductive parts, which so not male man, the female reproductive parts which are actually involved in the sexual reproduction. So what is the actual thing that we need? We only need this thing for the dissection of ovary. So just cut all of these. Okay, cut to this thing. Okay, we do not need all of these. Just cut all of those, and finally we are gonna get this thing. Okay, we are gonna get this. Fine. So now our duty is to do a very small. You need to be very skilled when it comes to this. Okay. Slowly, you just need to cut a very, very thin pieces possible. Okay, very thin pieces. This is thick. What I'm showing you is thick. It should be very minute pieces, guys. Okay, because it's more nicely visible. If you're how much thin is your pieces, more nicely visible. It is under the microscope, so you just need to cut a very thin pieces. Okay, and fill a watch glass with the water. Fill a watch glass with the water. Fill a watch glass with the water, guys. Yeah, this will be filled with water. Just cut the small pieces and put it into this. Okay, you're gonna get some small pieces. Yeah, you're gonna get some small pieces like this and all. Put it inside the water, and then with your beautiful and a very good eye, just search for a very thin piece. Okay, search for a very thin piece. Select it. Do nothing. Just take a glass slide. Select a very suitable one. Keep it under a glass slide. If this is a glass, just keep it under the glass slide. Okay, and you know what? We'll even put water in the glass slide and just cover it with a cover slip, and you're good to go. But here the different thing is that you're using a simple microscope, not a compound microscope. Okay, you will be looking this under the simple microscope. So this slide would give you 3 marks. Okay, this slide would give you 3 marks. Fine. So this slide will give you 3 marks. So where are you getting the remaining 2 marks from? You're gonna get it. One thing you need to report. Okay, you're supposed to report what are the numbers of the locule that is present inside that. So speaking about the hibiscus family, let me just show you how is it going to look. Okay, under the microscope. This is an ovary. There would be this 5 divisions. 1, 2, 3, 4, 5. See, this would be there in a more better way, but I would just want you to ignore the drawing for now. Yeah, 1, 2, 3, 4, 5. Yeah, that's all. There are just 5 things, 5 locules. So yeah, you need to report that it is a pentalocula. And then you need to report what is the type of a placentation. Okay, you see the ovules are arranged in this way. Okay, yeah, the ovules are arranged in this way. So obviously this is our axile. Axile placentation, and this would also give you 2 marks. Okay, and this would give you your 2 marks. So totally you're going to get your 5 marks only by doing so much. So this is our third experiment and the last experiment. Moving on to the last experiment. And finally to the last one, this is a nucleic acid staining experiment. Nucleic acid staining experiment. Okay, so what are we doing here? Okay, this is only a thing where we're using something other than the flower. Onion, guys. We are using a onion. Yeah, we are using a onion. So what are we doing? We're just taking a small piece of an onion. Okay, it would somewhat look like one small slab. Okay, and there would be a epidermal layer over here. Okay, there would be an epidermal layer, this thin thing which I'm drawing, right? So this would be your epidermal layer. It's a very thin stuff. You just need to cut open this by using anything, your hand, yeah, right, your hand or your wish, right? You just need to cut open this thing. Yeah, once you get it, you're gonna get a very thin layer of our onion peel, epidermal layer of the onion. You're gonna get a very thin this thing. You're supposed to place it under the glass slide. Supposed to place it under a glass slide. Okay, place the supper onion thing under a glass slide, and then which is the stain that you're using here. The stain that you're using here will be our acetocarmine. Okay, acetocarmine. So the stain which you're using here is the acetocarmine. We are using the stain. Here we are using the stain which is a acetocarmine, and place it under that for like a pretty much few, a bit amount of time and all. Keep it under that. Okay, until it's properly stained, until this thing is properly stained, keep it under that for a few times. So once it is properly stained, right, take the another glass slab where you put this thing, okay, where you put this acetic acid. No, acetamine, just keep it over there. Acetamine, you put the acetamine, and yeah, you're gonna get it. The small slab, just keep it over there, and then cover it with a cover slip. Okay, after keeping it for like 5 to 10 minutes, cover it with a cover slip, and you're good to go. Show it under the compound microscope. Okay, show the slide to your teachers under the compound microscope. This is gonna get you 3 marks. Okay, you're gonna get this 3 marks out of this. So now again, you're getting those 2 marks by reporting the shape of a cell. So yeah, I just show you how does it look. Okay, this is how the shapes of the. This is how the shape of onions will be looking under the microscope. Okay, it's a rectangular shape, and it will be having only one nuclei. So for reporting that it's rectangular shape, you're going to get 1 mark, and for reporting that it is uninucleate, it has only one nucleus. Okay, first of all, rectangular shape, rectangular and uninucleate. Yeah. So yes, you're going to get 2 marks because of this, and yeah, you're also done with your 5 marks. Totally, you're going to get your 5 marks finally. So that's all about the class 2 arts practical first question. Again, this depends on your luck, and since this is very easy, I would prefer you to not skip anything and just don't learning anything, don't test your luck over here. But if it does not come to you, you're going to lose like 5 marks. It's not accepted, right? So learn everything and go. Yeah, always based on what you want, you will be getting this by the lottery way. You're going to get something by the chit you pick, you're going to get some experiment, and you're going to be doing it. It's very much easy, nothing to be done, and you're easily going to get 5 marks out of this. Okay, this is all about the question number 1. Thank you so much, guys. Bye-bye.